So we are on, sorry, it's chapter 18, not 19. So chapter 18 in the free textbook is on blood. So most of the pictures I'm showing you, I just took it from online. Most of them are from SlideShare. I know some of you have the Martini textbooks, so the pictures might match. So we'll go over so the learning objectives is we are studying blood, life-giving blood. If you lose blood, after a certain extent, it will affect homeostasis, and then it does lead to death. So blood, you have to study the chemical composition of blood. You have to identify all the proteins, electrolytes, and solutes that are hanging around in blood. We'll talk about the liquid part of blood, which is plasma, and the solids in the blood. The solid in blood is called formed elements. So we'll study those. And we'll study how RBC is made in our body. We'll study how white blood cells are formed in the body. And one important application of this knowledge is blood typing. Several of you would have donated blood, and it's life-saving, right? So, we might lose blood because of maybe a surgery or an accident. So, blood bank is always looking for blood. So we'll talk about the science behind blood donation and briefly talk about blood disorders. First thing to remind ourselves is we studied in AP1, blood is classified as liquid connective tissue. So, it's strange to think of a liquid as tissue, but blood is. So it has a liquid matrix, it's called plasma. And then in the liquid, we have specialized cells that are floating in it. So the main functions of blood, which is important, is first is transport of nutrients and gases in our body. So blood, through the internal network of arteries and veins, the blood is reaching every cell in your body, provides the oxygen that is required for cellular respiration. Because if there's no oxygen to the cell, there is no cellular respiration, there's no ATP, and that leads to death of the cell. So it is vital that blood takes the oxygen to the cell and removes the waste, which is carbon dioxide. So prime function. A cell cannot survive with essential nutrients and electrolytes. Again, it depends on the blood. The endocrine system hormones that we studied that affect the activity of the cell is brought to the cell through blood. So it plays a huge role in the transport of gases and nutrients and all the things required for the cell and removing the toxins from the cell. It plays a huge role in homeostasis in our body. So blood regulates the pH of all the interstitial fluids in your body. It regulates your body temperature. It keeps you warm. And then it plays a role in water content, making sure there's enough water in your body. It is also very protective in function because it doesn't allow much blood loss and liquid loss from the body by triggering clotting in the body. And all your antibodies are hanging around in your blood. So it plays a huge role in disease prevention. So when, so the white blood cells, they are like policemen, they're hanging around in the blood. And if a bacteria or virus should enter your blood, they are the first line of defense. So in short, blood plays a huge role in transport, homeostasis, and it is very protective in nature. Some physical characteristics of blood. Blood is neutral. So the pH slightly alkaline, neutral, more than neutral. It's 7.3 to 7.4. This is the ideal pH of blood. It's slightly warmer than the body, 38 degrees centigrade. It's slightly thick. It is 3 times thicker than water. So that's why, like, if you had ever felt blood in your hand, you had a cut, and then you would feel it sticky and thick. So it has a higher viscosity. The color, So like if you're light skinned, you'll be able to see arteries and veins in your hand. And then the arteries are bright red in color because they're carrying oxygen. Whereas the veins are like purplish, bluish kind because they are deoxygenated. So that's with the color of blood. And the amount of blood, it varies with adult male and female. Men have more blood. So the blood volume in males is, adult male is about 5 liters, and then slightly lower in females. The hemoglobin also is slightly higher in males. So blood is a vital part of your cardiovascular system. So when you say cardiovascular system, there are 3 main components in your cardiovascular system. Of course, it includes the heart. We will study the heart next week. It's a double pump. It's a pump, 2 pumps side by side. So the conducting system refers to all the vessels that leave from the heart. So the supply that goes, the blood supply that's going to the lungs to pick up oxygen is called the pulmonary circuit. So after it picks up oxygen in the lungs, it will come back to the heart here. And then the heart will pump the blood to the rest of the body. So that is called systemic circuit. So the cardiovascular system comprises of the heart, the conducting system, and the fluid that's flowing inside the arteries, which is called blood, which is classified as fluid connective tissue because it has a fluid matrix with specialized cells in it. So here, there's a picture of a common procedure that's done in hospitals here. So you might have given blood, so you are probably familiar with it. Blood is a reflection of everything that's happening in your organs and tissues. So it is a very powerful diagnostic tool. When the doctor doesn't know what's wrong with you when you are sick, one of the things they are going to ask is blood. And then they'll analyze the blood and they'll find out what's wrong with us. So here's the arm. So if you are a nurse and you are familiar with this, so the blood is drawn and then they centrifuge it. That process of separating the liquid and solids in blood is called fractionation. So here is fractionated blood. And you can see plasma, or the liquid part, is a straw yellow colored, and it's about 55% of the blood. And then everything that settles are the solids, and they are called formed elements. They are approximately 45%. And then in between is where you would see the white blood cells and platelets. So they are called buffy coat, the white part. So here's a histology of blood. It's a blood smear. And in the blood smear, you can see the pink balls, it's stained that color, are red blood cells. And you can see the larger cells. It has this weird nucleus inside, the multi-lobe nucleus. They are the white blood cells. So they are the ones that fight against immune system. They are the immune system of our body fighting against foreign invaders. And you can see the third solid called platelet. So you can see the relative size and proportion of each one of them. So here we have about 5 million red blood cells, are also called erythrocytes. 5 million per millimeter cube of blood. Whereas if you take WBCs, there's only about 7,000. These are all approximations. So the approximate ratio of red blood cell to white blood cell is about 1 in 600. Or sometimes it's 1 to like even 1,000 sometimes. White blood cells are called leukocytes. Leuko means white, cyte means cell. Platelets are also called thrombocytes. So the formation of blood, they'll call it erythropoiesis. For white blood cells, they'll call it leukopoiesis. Poiesis means to synthesize. Or thrombopoiesis, to make platelets. So platelets play a huge role in blood clotting, which is essential because we might get hurt, and it's the blood usually clots and it seals so that we don't lose blood. So let's talk about each one of them in detail. We already studied all this, the physical characteristics of blood. Adults will have, male adult has about 5 to 6 liters of blood, and females have about 4 to 5 liters of blood. And then once you draw the blood and fractionate it in a centrifuge, you can see what is found in each one of the layers. So if you take plasma, which is approximately 55% of the blood, in turn is made up of mostly water, 7% proteins, and 2% of like electrolytes in it. All your calcium and magnesium are hanging around in it. All your hormones are also transported in the plasma. The 3 main proteins are albumin, globulin, fibrinogen, that plays a role in blood clotting. Albumin is very important for the maintaining osmotic pressure of blood. Globulin, that word comes from immunoglobin. So that refers to the defense mechanism in your body, antibodies. Solutes can be ions, all your nutrients. You had a healthy breakfast, and in all your glucose and amino acid, everything is hanging around there. The other solutes can also be waste products like urea, lactic acid. Your body wants to get rid of it. So the blood will carry it either to the kidneys or your intestines to get rid of it. There's also gases there and some hormones there. Buffy coat is made up of, there's about 5 to 10,000 white blood cells per cubic millimeter. So this is a very, good picture for y'all to understand and memorize. Platelets, only 200 to 400 per cubic millimeter. Platelets play a role in blood clotting. White blood cells is immune system. So if you zoom into the white blood cells, there's more subdivisions in it, like neutrophils, eosinophils, basophils. So we'll study all that in the future slides. But this is almost kind of like a intro slide as to what we'll be studying now.
Professor, I have a question.
Yes?
Do you want us to know all those percentage?
Approximately, yes. Is this Amanda?
It's Miranda.
Miranda. Yes, Miranda. Yeah. You have to know at least approximately, the pH. I know there's lots of numbers. You'll have to know that plasma is 90%, approximately. Actually, all of these are ranges. I didn't want to show it too complicated. That's why I removed the range. Yes, you have to know.
Okay.
Because these proportions play a huge role in homeostasis in blood. Anytime this proportion is upset, right? Like for anemia, your red blood count is decreasing, and then that's manifest as a huge disease. All right, plasma is a very busy liquid here. It's one of the most important body liquids. So it's 55% of the blood, mostly water. 91% is water, that's showing you how important water is. And then it has higher concentrations of nutrients and oxygen when you compare it with interstitial fluid, the fluid that's between the cells and outside of the cells. So relatively, it has more oxygen and proteins. So where do all the proteins in the plasma come from? So we know all the hormones in the plasma came from the endocrine system. We saw this last class. So now I told you there's this albumin and all of those globulins. So where do they come from? They are mostly synthesized in the liver. The most important protein is albumin. I'm thinking of eggs, it's so rich in albumin. And our blood is also so rich in albumin. So the most important thing about albumin is it plays a vital role in maintaining the osmotic pressure of blood. So that's why blood is viscous, and it's so important. It has to be the right viscosity. because that will keep water in the blood. It is maintaining the water balance of the blood. That is like related to blood pressure and all that, blood volume. They also transport fatty acid and steroid hormones, like thyroid hormones. Globulins are 38% of the plasma protein, and they are the antibodies. They're also called immunoglobulins. And other functions for them is they can carry some vital metals. So they're called metalloproteins, or they can carry lipids or lipoproteins. They can also carry steroids. So they can also act as transport proteins. Part of your plasma is fibrinogen. And where does fibrinogen play a role? It plays a huge role in forming fibrin threads during blood clotting. So it's very important. And here's the list of all the hormones that we studied last week: insulin, prolactin, thyroid stimulating hormone, follicle stimulating hormone, luteinizing hormone. All these hormones made by the endocrine glands are secreted into the blood, and it is the blood that will take the hormone to the target organ. When they remove the clotting factors from the blood, it's called a serum. So that's the difference between plasma and serum. All right, see, this picture might look complicated, but it's not very complicated. It's talking about, see, when we studied ANP1, y'all studied, where is blood formed? Where in our body is our blood formed? In adults, right? We knew it's formed in the bone marrow, red bone marrow of long bones. So in the bone marrow in adults, there are these stem cells. So a stem cell is a cell that can differentiate to different types of other cells depending on chemical stimuli. So the stem cell here is hemo. Hemo means iron containing group, heme, iron, cyto, cell. Blast means to actually make. So hemocytoblast is the mother cell or the stem cell. So now you can see, depending on the stimulus it gets, it can either differentiate into a pro-erythroblast. And again, that goes through a series of differentiations to form red blood cells. It can differentiate to myeloblast, and that becomes all the white blood cells, which are called granulocytes. So they have these granules of enzymes in them, and they stain like granules. That's why they're called granulocytes. So the myeloblast takes this lineage. Lymphoblast differentiates to form lymphocytes. The monoblast differentiates to form monocytes. They don't have granules. So when you stain them, you don't see dots there. So they are called agranulocytes. So these are all fighter cells, defense cells, all these. There's another differentiation of the mother stem cell to make a cell called megakaryoblast. And that will break up into small pieces, like pieces of plate. Maybe that's why it's called platelet. That is responsible for blood clotting. So again, y'all have to know the first differentiation and the last here. These two are important. You're going to see this repeatedly. We'll study this again when we study the immune system. So this is how blood is formed. Blood in adults is formed in the red bone marrow through the mother cell called hemocytoblast. That will differentiate to all these lineages depending on the stimuli. Let's say your blood count is low, your RBC is low. It can happen during several disease conditions, right? We've heard of stories of how the RBC count drops, in some, even cancer patients. So what happens is when there's less blood, erythropoietin is the hormone made by the kidneys, and then it will trigger the stem cell to take this lineage to form more RBCs. So let's look at the most important molecule in the blood. So what is the most effective shape of red blood cells here? So you can see, I really hope you had seen this in the microscope. It's like really beautiful. It looks like a little donut, bright red donut. It has a depression in the center. So this unique shape, like if you take the cross section, you can see how the red blood cell, which is about, 7.2 to 8.4 microns diameter. So it has a thick outer ring and then it is thin in the center. So now, why is this unique shape important? Because this unique shape allows for high surface to volume ratio. We can pack up a lot of hemoglobin protein molecules in there, and then they can carry oxygen. So the thing is, the RBC is only as wide as the smallest capillary in our body. They can like stack up on top of each other, and that formation, they can stack up on top of each other and they can wiggle their way, they can bend and they can maneuver through all the fine capillaries in our body. It's called rouleau. See that word, rouleau. So hemoglobin is constantly made in our body, every minute in your body. Like we make millions of them. And they live only for 4 months, 100 to 120 days. We make about 1 to 3 million RBCs per second. This is huge. Because that's the demand for oxygen and nutrients is so high. So the next question you should ask is, okay, they're living only for 4 months. What is happening to them? So they began, we saw the beginning in the bone marrow, and then we are saying it ends in 120 days. So you should be thinking about how RBCs are recycled. Before we understand that, we have to understand the molecular structure of hemoglobin. See, the discovery of the structure of hemoglobin and how it works, we're all very huge in biology. So the hemoglobin molecule, see, the RBC is packed with the molecule, the action molecule called hemoglobin. There are 4 strands. So there are 2 alpha strands shown in purple here, and there are 2 beta chains. So in the quaternary structure, you can see there are 4 strands. In the center of every strand, there is this unique chemical structure called heme. It's called heme group. And right in the center of the heme is where we find iron. And it's this iron. Iron is also called, this is the heme group. So this iron is vital because a hemoglobin molecule is also called as an oxygen raft. Oxygen raft. So it can carry oxygen. That means it's like a magnet. It attracts to oxygen. When it's oxygenated, it is called oxyhemoglobin. And then when the concentration of oxygen is less in the tissues, it will dissociate from the blood and enter the tissues. Then it is, when it's deoxygenated, it's called deoxyhemoglobin. So the thing is, it can toggle between these 2 states of being oxygenated or deoxygenated. So you can see the 4 strands, 2 alpha and 2 beta strands. So when blood goes to the tissues after it supplies the oxygen, the waste or carbon dioxide content in the tissue is very high. So what happens is the carbon dioxide will jump in and occupy, it will bind with the heme group. So when carbon dioxide jumps in at the tissue level, it is called carbaminohemoglobin. Very soon, when you study the respiratory system, you will study that the waste carbon dioxide in your tissues are carried in 3 ways. Hemoglobin is just one of it. It can also be carried by being dissolved in the plasma or as a bicarbonate are bound to amino groups in the hemoglobin. So there are 3 ways carbon dioxide can be carried in blood. So, like menstruating girls, like anemia can be, it's quite common in teenagers. So anemia is a condition where there is not enough oxygen in the blood because of a reduced number of RBCs or something going wrong with this affinity of carrying oxygen. All right, so every 100 to 120 days, that's the lifespan of our RBC. So what is happening at the end of 120 days? Why is it so short-lived? So it is, see, like it's one of the most unique cells in our body that doesn't have a nucleus. It doesn't have any organs. The ATP that it requires for survival comes from anaerobic respiration. Because we do not want RBCs to have mitochondria in there that is going to compete for the oxygen. Instead of supplying oxygen to the tissue, if there was mitochondria in here, it is going to compete for that oxygen. So that's why they are short-lived. So the breakdown and recycling of RBCs or erythrocytes is called hemolysis. And again, the story of recycling is very interesting. So what will happen is, see, I just showed you the structure of hemoglobin. It has the 4 proteins and the heme group, right? So macrophages that are found, so the spleen is the graveyard. Spleen is the recycler of RBCs in your blood, main organ. Also some lymph nodes and liver. But so macrophages will break down the hemoglobin molecule to heme and globin. And then through a series of reactions, it will be broken down to something called biliverdin, which is green in color. And then that is further broken down to bilirubin. And then it is modified in the liver. And so there's bilirubin in the bile. So that is a thick yellow liquid. And then it is gotten rid of through your digestive system or your kidneys. That's the reason as to why your pee is pale yellow color and our poop is also yellowish brown in color. So that's how that is gotten rid of. Let's see, so the globin is a protein, right? So that will be broken down to amino acids, and the amino acids are recycled for use for making other proteins. The iron in the heme, there's other proteins called, ferritin, transferrin and all that. So they will take the iron back to the bone marrow where it can be recycled, put back into making new hemoglobin molecules. So nothing is wasted. Nothing is wasted. So when RBC breaks down, it is completely used up. So how do we regulate blood formation? So the blood formation is called erythropoiesis. Erythropoiesis. Hypoxia refers to a condition of low blood oxygen. So when your body detects that there's not enough oxygen in the blood, see, there are receptors in the kidney. Hypoxia, inducible factor in the kidney, is constantly monitoring blood oxygen levels. As soon as it picks up the signal that there's not enough oxygen. So think about situations when your oxygen level might drop, right? I don't know if anyone has climbed a mountain. So like if the altitude is high, like Sierra or Kilimanjaro, or like Peru, like high, like Himalayas. So the higher up you go, the oxygen content is low. And it affects some people very much. Some people are not affected too much. But what happens is when the oxygen level drops, there are receptors in the kidney that will pick up this very important message, because it's going to affect homeostasis. So it will produce a hormone called erythropoietin, EPO for short. And then EPO will work on the stem cells, and then it will trigger erythropoiesis, thereby increasing the RBC content. So that's what is written here. So erythropoietin is made by the kidneys. It will go to the bone marrow, and it will ask the bone marrow to make more RBCs. And when RBCs increase, the oxygen content is restored. So that's why, going back to the climbing a mountain analogy, usually when they are climbing mountains, the climbers are asked to acclimatize. You know, they'll go to a certain height and they will just spend, like, if you're climbing Mount Everest, they have to acclimatize for at least 6 weeks. So they're waiting for this to happen, so that they'll be able to handle low oxygen levels. The picture on the right here is showing how the stem cell is making RBC. So this is showing a series of differentiation. So you can see how the mother cell for RBC is quite big. See, pro-erythroblast. And then day 2, it has differentiated to become a basophilic erythroblast. And then day 3 and day 4. But between days 5 and 7 is when it gets shaved off. See, the nucleus is pushed out in a week before it becomes a red blood cell and gets its unique shape. And as I told you before, that donut shape and the size of 7 microns is very, very important, so that it can carry, you don't want the nucleus to occupy space. We want the protein to carry oxygen, hemoglobin. And then you need that specific shape so that it can, like, wiggle its way through the capillaries. All right, this is very interesting. You would have probably heard about blood typing. So the main blood types are, you can either be an A, B, AB, or O, right? And then you can either be positive or negative. So that rhesus factor discovered in the monkey first, a rhesus monkey, it's also called D. So what is an antigen? So red blood cells especially, they have surface proteins, which is a glycoprotein. It is a glucose molecule and a protein together. It's like the signature of that cell. See, it's marking that cell. It is called as a surface antigen. So when in immunology, an antigen is a protein or like, it can be anything, like a glycoprotein, a protein that will induce your immune system to make an antibody. So that's the definition of an antigen. An antigen will evoke the production of an antibody. So when you say you are blood type A, you have A antigen on your RBC. You see, the picture is showing it. If you are B, you have B antigen. If you're blood type AB, you have both of them. And if you are O, you have 0 antigens. So if you are blood type A, you will have B antibody. So see, it's shown in B antibody. B will have A antibody. So that's why you can't give blood B to a person with type A. So during blood transfusions, you have to think about compatibility. So one more time, type A has antigen A on its phospholipid bilayer on the surface. So it is like a signature of that blood cell, and it's genetically determined. So if you're having antigen A, it doesn't make sense that you'll have antibody A. You'll have antibody B in your plasma. So that's the reason you cannot give blood. A A-type person cannot be a donor for type B, because if you mix these together, it will clump, and that clumping is called agglutination. So there are specialized people for this. So after you donate blood, and there are people, hematologists who are specialists in this. So for a blood transfusion to be successful, the blood types and Rh factor must be compatible. If they are not compatible, they will clog or clump, and that is called agglutination. So this is a picture of agglutination. So see, so here is type A blood that is having B antibody. So in its own blood, there is no agglutination. But when it's mixed with a type B, they will all come together and it will form agglutination. So this is a chart that you can study that talks about what blood type is compatible with what donor here. So you can see if you're O negative, you are a universal donor. Everybody can accept your blood. Whereas AB person, type O is the universal donor. People with AB blood are called universal recipients. They can take blood from A, from B, from AB. So they are the universal recipients. If you're Rh positive, people with Rh positive can get blood from either positive or negative. Whereas if you're Rh negative, you can only get blood from negative. So that's, that's a lot of info about blood cells. Kindly watch several lecture videos. So if you go to YouTube and you put blood lecture, I was watching one from educator.com. It was very good. So if you don't like the videos I have posted, please look for videos on your own and, master the subject. Okay, moving on to white blood cells. They are also called leukocytes. So they are the protection of your body. They are protecting your body from bacteria, from viruses, any foreign invaders, pathogens. So they are going to patrol the blood supply here. So you can see them. But one important, interesting fact about white blood cells is they will leave the blood supply and they will enter the tissue and interstitial fluids, and they will patrol the tissues also. So if you have like a paper cut and then a bacteria got into your tissues, the white blood cells are waiting there. They are like one of the main lines of defense in our body. This movement is called diapedesis. We have about 5,000 to 10,000 white blood cells per millimeter cube. They have, so when they move, they kind of have amoeboid motion, and they are phagocytotic. As soon as they see an invader and they know this is not a normal cell, they will engulf it, break it down. So that is called phagocytosis. And they are attracted by chemical stimuli. Certain cells, when they are affected, they will give out chemical signals. So these WBCs will respond to that stimuli. They are slightly larger than the white blood cells. They have a huge nucleus, unlike red blood cells. They are also produced in the bone marrow, and they will hang around in your blood for 10 hours up to years. So there are two types of leukocytes depending on certain proteins that will show up during staining. So granulocytes have granules in the cytoplasm. Agranulocytes do not have granules in the cytoplasm. And here's a good mnemonic to remember all the different WBCs. So in the buffy layer, you see leukocytes and platelets. So when you take leukocytes, 70%, most of them are neutrophils, and they are granulocytes. Another category is called eosinophils, that is also granulocytes, and a very small amount of basophil. So there are 3 types of granulocytes. And there are 2 types of agranulocytes, which are lymphocytes, which is a lot. See, the 45% is lymphocytes, and then monocytes. So we have 3 types of granulocytes and 2 types of agranulocytes. All right, this table is an important table. So here, under white blood cells, There are three types of granulocytes. Neutrophils are the most important. Nearly up to 70% can be neutrophils. What is their function? They phagocytize microorganisms. So these are the one they are going to attack the bacteria and virus and destroy it. Basophils. So you can see the multi-lobed nature here in neutrophils, and all the dots are these granulocytes. So here in basophil, this also have two distinct lobes. It will have cytoplasmic granules, and this is involved in your inflammatory response. See, they release histamines that will lead to inflammation, and they also play a role in blood clotting. Eosinophils. So they also participate in the inflammation response, and they attack and take care of parasites in our body and worms. So you can see the bilobe nucleus. So you can see how each white blood cell has its own distinct anatomy and physiology, each cell. Agranulocytes are your huge immune cells. They are the fighters in your body. See here, lymphocyte. See, you can see this huge nucleus. There is no lobes in this nucleus. It's a lymphocyte. So these are the ones that make all the antibodies in your body. And see, it says they contribute to allergic reactions. They take care of cancer cells, and they regulate the immune system. Monocytes, it has a huge curved nucleus here, and it's phagocytotic. It is macrophage. It is going to eat all the bacteria, dead cells in your body. So these are the examples of agranulocytes. These are the different types of WBCs in our body. So we would ask, all right, we studied how red blood cells are made in our body. So we'll see how white blood cells are made in our body. This picture is also important because we are going to be looking at it again when we study the immune system in our body. So again, it all starts in the bone marrow where there's a mother cell, which is undifferentiated. Depending on the stimulus, it's going to take different lineages. So I've already shown this before. So myeloblast is going to make all the granulocytes. Monoblast is going to make the monocyte, and lymphoblast is going to make the lymphocyte, which are agranular. The third farm element is platelets or thrombocytes. They play a huge role in blood clotting in our body. So anytime there's damage to the arteries, they will form a blood clot and they will seal it so that you don't lose blood. They are circulated in our blood for 9 to 12 days, and they are recycled in the spleen. So here we have a megakaryocyte that's going to make all these blood platelets. They're always found in blood and they trigger blood clotting. So the formation of platelets is called thrombocytopoiesis. Megakaryocytes are the giant cells that are found in the bone marrow that will differentiate to make the platelets. So here's the lineage. Megakaryoblast, it will undergo a series of differentiation, and then it actually bursts to form all these platelets. You can actually see them in the histology, in the slide of a blood smear. All right, the process of blood clotting is very complicated. There's lots of factors, they could call it intrinsic factors and extrinsic factors. So there are three main phases. So here we have an artery, and then because of some injury, it got cut. And the first thing your body has to do is to stop that blood from flowing out. So it's called hemostasis or cessation of blood. So there are three main phases in blood clotting. There is a vascular phase. So during the vascular phase, the diameter of the blood shrinks. So there is a decrease in diameter. And then all the cells, epithelial cells, will send out a distress cry, oh, there is an accident here, we are all broken. So they're releasing chemical factors. So those are happening in the vascular phase. And then in response to the factors, the platelets will rush to the site of injury here. They will all accumulate here and they'll form a platelet plug. This will trigger all the clotting factors, intrinsic and extrinsic clotting factors, and they will form a temporary patch to avoid blood loss. This will trigger real coagulation of blood. So there are many proteins. See, the fibrinogen in the blood that we learned earlier will be converted to these long protein molecules called fibrin, and they'll form like a network. And in the network, it will trap all the proteins and platelets, thereby forming a nice tight seal. So the procedure is written here. Platelets and damaged tissue will release clotting factors. Factor 8 is a very important factor. And then it will stop the bleeding. So here's a good video for you to watch that will narrate the different steps of how a blood clot is formed. So this is the last slide. So if you talk about blood disorders in the world, there's so many of them. You know, like you must have, hemorrhage is one, you know, internal hemorrhage is a bad word, and you might have heard the word thrombosis, you know, when the blood clot is like moving around in your body, or embolism. Leukemia, that is when there's less blood in the body. So when there's less RBCs in the body, the oxygen carrying capacity is lost. Sickle cell anemia, which is genetic, because of a point mutation and one amino acid chain, one nucleotide change in the DNA, instead of glutamate in the protein, you end up with valine. And because of that, the RBC kind of collapses on itself and it takes this sickle shape, which is very sticky, and it can clog arteries and capillaries. So, you know, it's very common in certain races. You know, it's common in India, it's common in the African American race. So this is a genetic disorder, sickle cell anemia. Leukemia. Leukemia is cancer here, leukemia. So there is a burst, there is an increase in the amount of white blood cell. See, again, going to show that proportion of all the different formed elements is very important. Hemophilia is when there is a genetic disorder, your body is unable to form blood clots and stop blood from leaving the body. So you can see how blood clot has, like, the blood is like, you know, gathered under the skin. Mononucleosis is caused by a virus. So they will attack the monocytes, and then it will have an enlarged nucleus. That's what's called mononucleosis. Lymphoma is also a type of blood cancer. You know, there's Hodgkin's, non-Hodgkin's. So these are the main types of blood disorders.